tell us about the seminar that you presented with regard to gene editing at OVO 2016.
So there was it's a course that we provided but a wide, diversified audience from ophthalmologists to PhD students. We started with the overview of different approaches how gene editing can be used in patient-specific stem cell lines, and then in mice and in other model organisms. And then we showed how to design the targeting, and also how to look for off-targeting of gene editing. In the preparation that we anticipate that in the next few years that therapeutic editing will be coming to human trials.
So to put us in context, in the last 12 months since we spoke, tell us about how these techniques have permeated and advanced.
I think one of the first, there's some proof of principle done in ophthalmology. There was in rhodopsin, they have been suggesting a rat model. You can use therapeutic editing to remove a part of the rhodopsin protein and have some suggestions of restoration of cell survival. And then another case is that you can also apply it to patient-specific stem cell lines in the carry defects in the X-linked form of retinitis pigmentosa. This was done mostly by Vinny Mahajan and Alex Basuk in pediatrics department at Iowa. That you can, with a pretty high 13% of the patient-specific stem cell lines can be repaired using gene editing. And then, of course, the next phase is the same kind of delivery system, the same kind of vector now is a proof of principle that likely also can be work in vivo in the human retina.
What are the implications for AMD, which is a polygenetic disease?
So you can envision, currently therapeutic editing is quite efficient to take out pathogenic genes or pathogenic alleles. You can, so it's the same as using an antibody or using the RNAi or antisense technology today to remove the abundance of pathogenic vascular endothelial growth factor in the neovascular wet form of macular degeneration. And then for the dry, non-exudative form of macular degeneration, the more than one target. But of course, therapeutic editing, you can do multiple targets, but in the short term, it's probably targeting a single target using by removing instead of repairing the genetic insult.
What are the vectors of choice that you're currently using and recommending?
There's a variety of vectors based on the specific case application. And you can just injecting the a ribonuclear protein complex. It works pretty well in to generate mouse models in mouse oocytes. So you can use it to now knock in or knock out a gene in mice. That works pretty well. It also works quite well in patient-specific stem cell lines. But in people's retina, it's not exactly clear what would be the best. Of course, for regulatory bodies like Federal Drug Administration, that the naked RNA and protein that would be the easiest to get approved. The next level is some kind of viral delivery system. You can use adeno-associated virus, or you can use a non-integrating lentivirus, or even adenovirus that can be used for therapeutic editing.
Could you comment on non-viral vectors such as the work that's being done at Professor Duda's lab with gold nanoparticles? What is the positioning and role for that approach?
So you're looking at complex the nuclease the used for gene editing, and also the donor template or the and then the ribonucleotide. You're going to be complex with a gold particle or complex with a liposome. You should be able to work with a non-viral gene delivery system. But the rate limiting step to bring it to the clinical use is delivery.
So if you look at the next year, what are your milestones that you'd like to see in your own research and in the research of your colleagues in this field?
Just in the last issue in Cell Stem Cell, Professor Pete Coffey showed that you can correct the patient's stem cells and correct some of the defect in terms of generating cilia in patients with early onset retinal dystrophy in the CEP290 gene. So you see a couple more examples of that that will work on patient-specific stem cell lines. There will be some more examples of corrections in animal models or generation of animal models. I think all this is to build up enough proof of principle data for human trial.
Could you comment on the human trials that have started?
Human trial has not started yet, but it has been said
Right.
there will be the first human trial will be started as 2018.
Right. Wasn't there some work going on in Europe?
This is the CEP290 gene. Has been there is a cryptic splice site in one of the introns, so the design is to remove the cryptic splice site in the form of early onset retinal dystrophy. So the Professor Coffey's group at Moorfields already shown there's a proof of principle in patient-specific stem cell lines, and there is as long as the delivery system work in people's retina, then the same thing can be helped in patients.
So finally, if you were to advise patients, tell people, lay people, what the horizon is, what the prospects are, and the positioning for gene editing in terms of their diseases.
But the previously unapproachable genetic disorder, for example, CEP290 is too big to be fit in conventional delivery system. And there are other genes as big as the CEP290, including ABCA4 and some of the usher genes. So those are previously daunting, but now it become feasible. And then previously also dominant disorder looks unapproachable, but now with gene editing, that is become feasible. And then for oligogenic or multifactorial diseases, including the dry form for age-related macular degeneration, that it's possible to now to decrease those genetic risk allele. And if gene editing proven to be safe, it can be offered to patients even before they have any detectable pathology.
If patients want to find out more about the work that you're doing, what website should they go to?
Just our Columbia University website, or columbiaeye.org.
And you're not currently recruiting patients into any clinical trials yet, or are you?
So the challenge for clinical trial is that we're now looking for, at this stage, gene therapeutic editing is to preserve people's vision. So we need to know the rate of change. So we now just we have to get a natural history, because most likely that we're not going to. The goal is not to improve people's vision, but to stabilize people's vision. So in order to see the rate of change, we need to get the natural history. So now we're just seeing patients with different genetic disorder to see how the rate of change, and then when FDA approved therapeutic editing or any kind of treatment, then we hope that the treatment will stop the current rate of change.
Is there anything else that you'd like to add?
So this is probably a little bit shift. For example, at this conference, before we have been describing a lot of DNA with the newer sequencing technology. because it used to take 15 years and $3 billion to sequence somebody's DNA, and now it takes literally it takes only 2 days, but then it takes a couple months of interpretation with a price of instead of $3 billion is $1,500. So we have been, and then with the great tools great strategies to sequence and observe how genetic change affect a diversity of disorders. So now, since the last few years, now we're beginning to using editing to not just observing genetic changes, but now we can now use it to repair and correct them.
A wonderful, wonderful vision. Thank you, Professor, for your efforts and your energy and your educating the next generation to make sure that we can all benefit from this incredible innovation. Thank you.